Yeah. Where's our collection hat? Where's the collection?
Got it right here. Oh no, he doesn't. No. Tasteful. Here are my things to keep you guys on time. I'm going to be a dictator.
Oh, he's a dictator?
Good, that helps. Are you going to be sitting right up here?
He is.
And you should just take questions until time's up.
Yes.
Oh, hi. Oh, it's our Yes. Thank you for coming.
Hey.
And you're
Chris
present?
Evans.
Oh, yeah. Okay, so I just...
When you don't like, we tested the system. If you just stand right here, it picks you up very nicely.
Okay.
Yeah, that's it.
Great.
So good. Yeah. If you, Dr. Fong, if you just stand right here and talk in this general vicinity, it'll pick you up really nicely. You don't need to like lean in or anything. So.
Which is good. And there's a lot of people here.
I know. Well, you know, it's sold out.
I know it's sold out.
Everybody's sitting in kind of apart, but they're eventually going to all have to...
It's also 9 a.m. Wait till like.
That's true. I mean, people will come trickling in. Yeah.
Do you think it's going to be...
We should start now.
Okay. Okay, so yeah, then why don't we get that?
Pretty good.
Well,
So if you can go for it.
oh, we got to speak in here?
Yeah, as long as you stand right there, it'll pick you up.
Okay. You sure?
It should be. Attention, everyone.
Okay, so I think we should get this all on the road. If everyone can take their seats and we'll get going. So I'd like to welcome you all. This is our first symposium on cannabis, and obviously there's tons of interest in cannabis now, which is great. This is really came out of an affinity group that the Brain Research Institute put together, and it'll be, this symposium is actually funded by the Semel Institute and the Brain Research Institute together, so we have a little emergence here, which is good. I'd like to first acknowledge the organizers, and Jeff Chen has really done most of the work here, so congrats to him. And Igor Spiegelman, I don't know where Igor is, over there. He's another organizer. Tim Fong and Sean Hussein, I think, I don't know where Sean is, maybe he's in the looking after patients, but he's going to be talking a little bit later about epilepsy and how he's had some very interesting findings with cannabidiol, so he'll be talking about it later. Also, I'd like to thank our speakers who crossed the country. Ken Mackie, where's Ken? Somewhere around, Ken's over there. And he's, he directs the Gill Institute at Indiana University. I've known him for many, many years. And Ziva Cooper, who's going to be talking more about the clinical aspects with regard to addiction. And so I'm not quite sure what she's going to be talking about, but she'll tell you. Okay, so that's, I'd like to thank them. I'm sure it won't have escaped you that this is 420. Now, I went to the internet to try and figure out what 420 actually was derived from. Turns out that it's Hitler's birthday today. I guess some of you probably didn't know that, but it is. Maybe something to do with the war on drugs. I don't know, but it is Hitler's birthday. It's also some sites it said it was something to do with a police code. 420 was a police code, but I think the most parsimonious one was that it was the time of day when these kids got together to smoke their weed after school for being 420. But that was it. Okay. What do you want?
Sorry, the system just cut out.
The system cut out? Uh-oh. Okay, I'll let Jeff fix it.
I think we're okay.
So, with Prop 64 really involving national relaxation laws on cannabis, you know, things are moving forward, and we're going to find, I think, a lot of there's going to be a lot more findings about what marijuana actually does. We're going to get the good, the bad, and the ugly from marijuana coming out in the next probably decade or two. So it's going to happen. The problem is, though, for research, it's really difficult because the scheduling of these drugs is still Schedule 1, so it's very difficult to do preclinical research and therapeutic research with these drugs, and I think that's something which we need to really focus on. So I should shut up now. I'd just like to thank everybody for coming. I think we should get on with the symposia. You don't want to hear from me, you want to hear from the speakers, and I'll hand it over to Jeff. So thanks.
Alrighty, thank you so much, Chris. It was Chris Evans. He's the director of the Brain Research Institute. And our first speaker of the day has actually flew in last night from Indiana, so thank you very much for traveling all this way. Is Dr. Ken Mackie. He's a professor of psychological and brain sciences at Indiana University, Bloomington. He's an adjunct professor of anesthesiology at Indiana University School of Medicine. He received his MD from Yale University and completed his anesthesiology residency at the University of Washington. For 15 years, he combined an active laboratory research program with clinical care and anesthesiology at the University of Washington. Dr. Mackie's research interests focus on understanding the relationships between cannabinoids, endocannabinoids, and the pharmacological and physiological responses to both. All right, give a warm welcome for Dr. Mackie.
Thanks, Jeff, for both inviting me as well as the kind introduction, and thank all of you for being here so bright and early in the morning, although for me it's lunchtime. So what I'd like to do today is talk, just to give kind of general background, introduction to endocannabinoid system, and hopefully serve as a framework for the rest of the talks today. My affiliations are there, and as well as my email, and if you'd like a copy of the slides, just send me an email and I'd be happy to compress them and send them to you. So as an overview, what I'll talk about is the, just sort of introduce endocannabinoids, endocannabinoid system, a little bit of history, some concepts that I think are important in understanding how exogenous cannabinoids, particularly THC, and other compounds from the cannabis plant interact with that, and then some open questions for the future. As for disclosures, my work is supported by NIH and IU Foundation. I've only consulted with Janssen recently, and no other commercial conflicts. In talking about the subject, often you end up between two extremes. One is that this is the worst plant that ever has come along, and anything that anyone touches, it leaves a place of life with depravity, versus cannabis can cure everything. And I think probably by the end of today, we'll see the reality somewhere in between those two. This is, again, what I'll talk about. So the endocannabinoid system consists of cannabinoid receptors, endogenous cannabinoids, and metabolic pathways. I want to introduce what a key role of this system is in mediating synaptic plasticity, and then talk about these guys. So as I'm sure 95% of you know in this room, what we're here for today is really to talk about cannabis and its many different forms. Important distinction is between cannabis that lacks THC versus cannabis that contains THC or related cannabinoids, and as you know, looks like this. Major psychoactive component is delta-9 THC. There's a number of other very interesting compounds in the plant, 60 or so that fall into the chemical structure of cannabinoids, plus many that are others like the terpenes. We'll hear quite a bit today about CBD, cannabidiol. Various preparations that affect potency and effects. Typically, raw dried plant is the starting material of the female plant. The THC is primarily found in flowers, leaves, and buds, stems, seeds, weed, roots, no good. And then one can take the dried plant material and process it in various ways to increase cannabinoid content, particularly THC content, such as hashish, or more sophisticated, as in extracting with either, organic solvents or CO2, and then drying that down, as well as various oil preparations. Route administration, classic, of course, is smoked through combusted material in various forms. More sophisticated fashion that helps avoid some of the detrimental side effects of actually burning something is heating to the point of vaporization. THC vaporizes at about 180 degrees Celsius, and of course there's lots of ways people do that these days. And then other route of administration that's actually quite interesting in that it subjects THC to considerably different metabolism, and what hits the brain is a little bit different is if cannabinoids are ingested in edible forms, and that's quite easy to do, THC being so lipid soluble, easy to extract and prepare in foods. So there's some discussion about really what is a cannabinoid. I just wanted to spend one slide on that. And I mean, initially, basically, the idea is that a cannabinoid is a plant, is a compound, a chemical extracted from the cannabis plant or related plants that produces the characteristic psychoactivity of cannabis. Later, that sort of was extended to compounds with similar chemical structures, which may or may not be psychoactive. Then there's a pharmacological definition as a compound that interacts with cannabinoid receptors and produces cannabinoid psychoactivity. And that compound may be naturally occurring or synthetic. And then building on this are some distinctions. Phytocannabinoids being cannabinoid compounds that come made in plants, of course. Endocannabinoids or endogenous cannabinoids, the compounds that our bodies make. And then as well as synthetic cannabinoids, which are obviously synthesized. So the endocannabinoid system itself really came about from a period of sort of intense activity around cannabis and understanding how it's work. And you know, I guess I'm showing my age here, saying sort of this golden age of cannabinoid research in terms of understanding how cannabinoids work, as opposed to a lot of observational studies which were done earlier. This led to the discovery of endocannabinoid system, and if you'd like to sort of anchor it with other systems, there's a lot of parallels between the endocannabinoid system and the endogenous opiate system. And so basically it consists of receptors, ligands, and the metabolic enzymes that either make or degrade the ligands. And just as a comment, this is a system that is evolutionarily highly conserved in all vertebrates, and pieces of it, particularly the endogenous cannabinoids and sort of a slightly different suite of receptors, exists in lower phyla as well. If we put names on this, what we have for ligands primarily is 2-arachinyl-glycerol and anandamide, abbreviated as AEA. The synthesizing enzymes, acetylglycerol lipase alpha beta, phospholipase Ds, ABDH4, etc., and degrading enzymes here. And then receptors, I've shown two of the most well-studied ones, which are CB1 and CB2 cannabinoid receptors, but there are a number of other receptors that endogenous cannabinoids can interact with under physiological conditions, including some of the TRP channels, some of the PPAR nuclear receptors, etc. So cannabinoid receptors we'll define here as proteins that interact with endogenous and exogenous cannabinoids. The best characterized are CB1 and CB2 cannabinoid receptors, which I'll just abbreviate as CB1 and CB2. And then there's the additional receptors, including some nuclear receptors that are very important in gene transcription, and certain endocannabinoids, particularly the anandamide types, interact quite strongly with those, and they also interact with TRP channels. It's important to keep in mind through this that THC and endocannabinoids can interact with many other cellular targets. And you know, as a sort of a pharmacologist, in vitro person primarily, you know, we can put enough of any compound in a test tube to see an effect. And a lot of the challenge is really figuring out what are biologically relevant effects that we can measure in vitro. So moving on to CB1, this is showing CB1 localization using a CB1 antibody in monkey brain. And immediately some highlights pop out. Fairly high levels in cortex with a posterior to anterior gradient. You can see, looking here, there's a laminar-like structure within the upper layers of cortex, and then relatively high levels in structures such as the amygdala, globus pallidus, and then relatively low levels of expression in white matter thalamus and a few other brain regions. If we look now moving to rat and look more look more closely at expression of the mRNA for CB1 receptors, we see this very characteristic pattern. This is recapitulated throughout the nervous system, where there's a few neurons that express really intensely high levels of CB1 message, that's shown in dark blue here. Then some neurons that express intermediate levels, shown in lighter blue here, less, and then some neurons, this would be dentate gyrus here, that express no CB1 receptors at all. So that kind of recapitulates CB1 receptor throughout the brain. No medium and high levels of expression. If we look at what this mRNA gets translated to in protein here, we see a rather characteristic pattern of what looks, at least in those of you close to the front of the room, can see is sort of a fine meshwork of fibers. And that's characteristic of axonal staining. So the vast majority of cannabinoid receptors seem to be located in axons and presynaptic terminals. If we want to see, whoops, sorry about that. If we want to see, is this all the protein we see here, is it coming from these neurons or from these neurons? Using knockout mice, we can delete the CB1 receptors from these highly expressing neurons all through here and leave it behind here. And if you do that, you see this kind of expression. So this is total lack of CB1 receptors, no protein, just background from cell nuclei here. If you knock it out of just these intensely expressing neurons, you see this kind of pattern. So very light level layers over CA1, CA3, and a heavy expression in the intermolecular layer of the dentate gyrus. So pretty much all of this, except for this little bit here, arises just from these interneurons there. So very, very, very high levels of expression in a localized set of neurons. This is showing the same thing. And the point here is that if we look at G protein coupling, so CB1, CB2, or G protein coupled receptors, if we look at their coupling of CB1, either from CB1 expressing glutamatergic neurons, CB1 expressing GABAergic neurons, we see this interesting result. So if you knock out CB1 from all GABAergic neurons, just leaving it first approximation glutamatergic neurons, you see much more efficient coupling as indicated by the higher bar here. If you do the converse experiment, just leave it behind in the GABAergic neurons, you see much less coupling here. So this basically says that the receptor coupled in these, CB1 in these cells couples much more efficiently to downstream signaling processes. And this may be important in terms of how these receptors involved in processes like epilepsy. And there's a wide number of studies that show even though there's low levels of receptor expression in these cells, that expression is quite functionally important. If we look a little bit more closely at where these receptors express, so this is a section through mouse hippocampus staining for CB1 receptors, and you see this classic dark lines with little puncta on it. It's axonal and presynaptic CB1 receptors. If you use super resolution microscopy and look at that closer, so this would be taking this cell body here, looking at one synapse onto that cell body here, blowing it up, you see this sort of classic pattern. Here's green postsynaptic cell, the red presynaptic terminal, red being CB1 receptors here. And then you can look at even higher magnification. The light blue dots here are as a marker for the active zone where the neurotransmitter vesicles are released. You see cannabinoid receptors are expressed all over that axon terminal. So high levels of these receptors, very localized to where they might modulate synaptic transmission. Is CB1 only presynaptic? No. We see most of it presynaptic, that's why we focus on it. But there's a series of nice studies from, primarily from work from Giovanni Marcano and Viet Lutz, showing that CB1 receptors are expressed in other cells. This is looking in astrocytes, as well as in interesting subcellular compartments, such as in mitochondria. So this is an area that really will need more work. Is showing that activation of those mitochondrial CB1 receptors by a synthetic cannabinoid here actually decreases ATP production. So the signaling pathways of CB1 receptors, the inhibition of calcium channels, inhibition of adenyl cyclase, activation of potassium channels, all suppress that activating CB1 receptors might decrease synaptic transmission. The receptors we just saw are localized to presynaptic terminals. and multiple studies do show inhibition of neurotransmitter release. Just to get you used to looking at some of the data, this is a typical experiment. So this is done using hippocampal slices from rats in this case, using patch clamp recording, so looking at the synaptic input onto a single neuron and then just applying drugs and seeing what happens. So this is set up so they're measuring GABAergic currents in CA1 and hippocampus. So this is under control conditions. You stimulate the presynaptic terminals. You see a release of GABA and an outward current. If you put on this synthetic cannabinoid, you see a decrease in the evoked current. And then if you put on a CB1 receptor antagonist displacing the cannabinoid, you see recovery of the IPSC amplitude. So this is a very classic response. Appropriate activation of these receptors decreases synaptic transmission. Moving to CB2 receptors. So these are in some ways the poor but very interesting cousins of CB1 receptors in terms of not as nearly as thoroughly studied. So these are most abundant on immune cells, including microglia, so nice high expression or pervasive expression in the brain on brain microglia. They're interesting in terms of their regulation if they're highly inducible. They may be present at low levels under normal conditions. After pathological insult, their expression level increases quite strongly. They may be expressed in some neurons, although this is quite controversial. This is illustrating some of these effects. So this is a multiple sclerosis model. You can see normally in the spinal cord very little CB2 mRNA. A few days following EAE induction, you see strong upregulation. This is on a log scale, so about 100-fold increase CB2 mRNA in spinal cord. And this is looking following a peripheral nerve injury by crushing the nerve and looking at either CB2 mRNA or a marker for activated microglia. You see after a few days upregulation of CB2 message in appropriate portion of the spinal cord that overlaps with expression of this microglial marker, so suggesting that CB2 receptor expression increases in spinal cord after injury. There's a long history of potential anti-inflammatory effects of cannabis and cannabinoid preparations, and some speculation CB2 receptors may be the receptors that mediate these effects. And there's a number of intriguing studies that suggest CB2 may be an interesting potential therapeutic target for a number of different disorders, pain, fibrosis, etc., that potentially avoid the psychoactivity of CB1 ligands. So moving to endogenous cannabinoids now. So the presence of receptors in the brain suggests endogenous ligands. Right? Remember, these receptors are expressed all through vertebrates, so even fish, which have not too much access to cannabis plants, express cannabinoid receptors. I did that a few years ago. One person in the audience pointed out, what about seaweed? So that was Vu at Cold Spring Harbor. Vu at Cold Spring Harbor. So two main families have been identified. In common, they're both arachidonic acid derivatives. They both exist as precursors in the membrane, so that they have in common. Otherwise, they're quite distinct. The first class are the amides, typified by anandamide. Structure of that's shown here, so basically just arachidonic acid amidated to ethanolamine. And then the second family are the esters, 2-arachidonoglycerol being the prototypic member there. So again, arachidonic acid, but here esterified to the 2 position of glycerol. However, there's significant differences. The routes of synthesis are completely different, so they'll be synthesized in the body under very different conditions. And then the mode of degradation are different, so they'll be degraded by different families of enzymes and regulated in different fashion. And then from a signaling point of view, their efficacy at both CB1 and CB2 receptors are quite different from each other, where generally 2-AG is a more efficacious agonist. So anandamide, again, this is a precursor, and this is the canonical pathway. So you have phosphatoethanolamine and phosphotylcholine, and through an N-acyl transferase, which is the rate-limiting enzyme of this step, you get transesterification of arachidonic acid from here over to here to give this precursor N-arachidonyl phosphatoethanolamine, and then through the activation of phospholipase T, this is cleaved, giving you anandamide. So the nice thing about this is you basically have your neuromodulator in the membrane ready to go. You just need activation of enzyme. Anandamide degradation is, in the simplest form, is quite straightforward. You have anandamide here. You have a enzyme fatty acid amino hydrolase that then cleaves it to arachidonic acid and ethanolamine. And interestingly, and this is a recurring feature in the endocannabinoid system, if you knock out this enzyme here, work from Ben Kravat's lab, you see strong increases in anandamide levels. So this is the wild type, and this is the knockout levels. However, you notice that this enzyme isn't around just to degrade anandamide. It also degrades other fatty acid ethanolamine, so palmitoyl ethanolamine or oil ethanolamine. Both are also degraded by this enzyme. So if you have either a drug or genetic either knockout or mutation that affects FA, not only will you increase levels of anandamide, you'll also increase levels of these other fatty acid ethanolamines, which may have effects on their own. Another feature for degradation of endocannabinoids, of anandamide, shown, so anandamide shown here, this is a classic degradation towards by FA towards arachidonic acid. It's also a good substrate for various cyclooxygenases, and basically through COX-2 we'll make a series of prostamides, and through thromboxane synthase we'll make thromboxane analogs. So another issue is if you inhibit degradation by this pathway, you potentially increase synthesis through these other pathways. So endocannabinoids have a lot of unintended consequences. And this is showing the most abundant of those, the PGE2 ethanolamide analog. And these signal through receptors that are not totally well characterized, but are not cannabinoid receptors. 2-AG synthesis, on the other hand, goes through slightly different pathways. So its precursor is phosphoinositol in the membrane. Following cleavage of inositol phosphate by phospholipase C, you get diacylglycerol shown here, and then a specific diacylglycerol lipase cleaves off this acyl group, leaving you just 2-AG here. 2-AG degradation primarily is through this enzyme monoacylglycerol lipase, and this is a activity graph shown here. So MAG lipase accounts for most of the degradation of 2-AG, but there is substantial degradation by a couple other lipases too, which have roles in specific cell populations. And they're localized in different places inside and outside the membrane, allowing hydrolysis inside or outside of the cell. One thing to keep in mind, especially with the next few slides I'm going to show you, is that we tend to think of endocannabinoid production in neurons, but endocannabinoids can be produced quite well by other cells. This is showing an example from Nepistella's group, looking at cultured astrocytes, having their purinergic receptors activated by ATP, and you can see large increases in this case 2-AG production by treating by activating astrocyte purinergic receptors. So particularly in pathological states, these signaling processes may come into play. This is just showing using RNA-seq. Neurons shown. This is RNA-seq from cortical cell populations. Neurons express modest levels of diacylglycerol lipase alpha. Astrocytes and microglia express quite substantial levels as well. And this is another thing that I think is important to keep in mind. So we tend to think of endocannabinoid, breakdown endocannabinoid. You inhibit that process, you have increased cannabinoid signaling. This is very nice work from Ben Kravat's group, showing that here, typically, you have 2-AG that's been made by diacylglycerol lipase. It's broken down by MAG lipase and gives you arachidonic acid. However, the arachidonic acid can go on and do other things as well, and particularly making prostaglandins. If you inhibit MAG lipase by a relatively specific MAG lipase inhibitor, JZL184, then you'll get enhanced CB2 signaling by increasing levels of 2-AG, but you have potential to also have decreased arachidonic acid signaling, which will give you decreased prostaglandins here. And this is shown nicely in an LPS model, so giving lipopolysaccharide strongly increases prostaglandins in the brain. So that's data here shown. JZL, LPS alone, no drug, no inhibition, no LPS, typical levels of 2AG, typical levels of prostaglandin E2. You give LPS, those go way up. You inhibit MAG lipase. Even in resting brain, you decrease PGE2 levels, but now LPS can no longer give those really strong increases in PGE2. This is nice, it's biochemistry. Does it have functional implications? And it does. So this is looking at striatum under control conditions or giving JZL alone, pretty identical. This is being stained with IBA1 to look for activated microglia. If you treat with LPS, you see the strong upregulation of IBA1, so activation of microglia. However, if you pre-treat with JZL before giving the LPS, this prevents microglial activation. This is not blocked by cannabinoid receptors antagonists, so it's not mediated by the increases in 2AG. Instead, it's mediated by removing the prostaglandin. So nice illustration of sort of the promiscuity of this signaling. Not only do you have to think about increasing the 2AG, but also what the downstream effects are. Okay. So summary of endocannabinoids. So they exist in the membrane as precursors as phospholipids. There's two main families, the acylethanolamides, that's an anandamide. They're quite diverse. They tend to be more promiscuous. There are many targets in addition to the CB1, CB2 receptors, TRIPS, PPARs. Acylglycerol esters, both are made on demand. 2AG is considerably more abundant than anandamide. You just grind up brain and measure the two. However, a lot of 2AG seems to be involved in metabolic pathways, and people hypothesize that there's similar signaling levels of these two compounds at CB1 and CB2 receptors. They differ in their efficacies. 2AG is more efficacious than anandamide. There's quite a bit of metabolic diversity in how they're made that I don't want to subject you to, but they're the core pathways that I showed you before. Inhibition of endocannabinoid degradation can elicit CB1 receptor and CB receptor and CB receptor independent effects. And so just as a visual summary of the processes, we have anandamide 2AG up here, CB1, CB2 is the tip of the iceberg. There's a whole lot more underneath the ocean in terms of both signaling targets and related lipids to keep in mind when thinking about the endocannabinoid system. So what do endogenous cannabinoids do? They're preformed in the membrane, liberated by activation of certain membrane lipases. These lipases are activated by neuronal activity in certain receptors. They're well positioned to function as feedback regulators of neuronal activity, produced by many different cellular elements in the brain. One is that endocannabinoids will inhibit synaptic transmission. THC, and the other hypothesis, THC elicits effects by interacting with this ongoing system in the brain. So this is showing one form of endocannabinoid-mediated synaptic plasticity. So here is the basic idea is you depolarize the postsynaptic cell, increase calcium, increase endocannabinoid production. Those endocannabinoids diffuse retrogradely to the presynaptic terminals that have the CB1 receptors, and they will inhibit neurotransmission. And that occurs in a process like this. This is recording from layer 5 of cortex, looking just at spontaneous inhibitory potentials. You put a depolarization on the postsynaptic cell, and you see a diminution of inputs onto that cell that lasts for tens of seconds. So this is a very nice retrograde feedback system. Can be the positive or negative feedback depending on whether this is an inhibitory or excitatory neuron. So it can either amplify the response or lessen the response. Being pharmacologists, we like to quantify things. So I want to show you this quickly as a way to explain the next slides. So this is typical electrophysiological recording. We record from postsynaptic cells, stimulate inputs onto it. We do that, we see this peak inward current here, and this is what glutamate activating AMPA receptors does. We do manipulation, we see a decrease in the current, and then that recovers. And we can quantify that by integrating under these curves, which will just give us total net charge transfer. We can do control, and then after treatment, and then just look at the ratio of those two. So we basically just plot these charged with time. And so if we're doing DSC, this is what it would look like. This is just our brief depolarizations. We get a EPSC. We can repeat that many times. If we then depolarize the cell here, the next EPSC will be smaller, and that recovers rapidly with time. And we can just plot the size of this post-DSC pulse with time and get something like this. So if we vary the length of depolarization and look at how small that next pulse is, we can see the pulse gets smaller and smaller the longer you depolarize the cell. So nice dose-response curve. So this is with cultured cultured neurons from not CB1 knockout mice that have CB1 receptors transfected back in. And these are if the neurons lack CB1 receptors altogether, showing it's a CB1 receptor-mediated response. There's several other forms of endocannabinoid-mediated synaptic plasticity. Some focus on synapses, and these are transient. This is long-lasting. And then there's one type that interestingly involves cell bodies and potassium channels. So quite a bit of diversity in these different processes. So summarize this. So transient and persistent forms of endocannabinoid-mediated synaptic plasticity. Almost all synapses that express CB1 receptors express some form of synaptic plasticity. They may only express one and not others. It's quite diverse. This is found in both excitatory and inhibitory synapses. So it's really difficult to predict net effects without knowing what the ongoing activities are. Because if you don't have any activity, you don't have anything to inhibit. So it's very, very circuit dependent and state dependent. Didn't have time to go into it. Anandamide can also elicit some interesting forms of synaptic plasticity through trip channels. And other cellular elements like astrocytes and such also likely contribute. So one question is, are the effects of cannabis due to THC's interaction with endocannabinoid-mediated synaptic plasticity? We think this is the case, but it wasn't quite the way we expected it to be. So one thing to mention is that CB1 receptor efficacy is quite variable, and efficacy basically means the maximal effect that you get out of the response. So this is looking at increasing concentrations of the drug here. This is maximal response normalized to the response of a synthetic cannabinoid. And you can see here 2-AG is fully efficacious, but methanandamide, which is metabolically stable anandamide, and THC are much less efficacious. So this suggests in this system, THC is actually acting as a partial agonist. So if we go back to our neurons and look at EPSC charge that I showed you before, put on a relatively high concentration of THC, we see no effect on EPSC. So it's not inhibiting like normal CB1 receptors would do. In contrast, if you put WIN on, you see this is potent, efficacious synthetic cannabinoid, you see good inhibition. And if you do a DSC experiment, this is the increasing duration depolarization, decreased synaptic transmission. You can see if you add THC to the mix, you actually antagonize that. So in this system, THC actually acts as an antagonist. So conditions favoring this outcome are those where there are either few receptors or low cheap protein coupling efficiency, and then conditions favoring agonism include high receptor density and efficient G protein coupling. So different synapses, THC will have very different effects. And so needs to say the net consequence of THC will be quite complex. And what we've hypothesized and certainly written enough about is THC's characteristic psychoactivity in part due to its low efficacy. The reason THC does what it does to your brain is because of its low efficacy. It's not just like activating the receptor everywhere. Some places will be activating versus endocannabinoids, other places will be antagonizing endocannabinoids. So what about spice compounds? So spice are these so-called synthetic marijuana. Basically, it's a contest between illicit chemists and the DEA for what's been scheduled. And it is possible to mine the very rich literature on CB1 agonists to synthesize compounds that will escape scheduling. How do these compounds affect synaptic transmission? This is one of the early spice compounds, JWH18. You can see, looking at EPSC amplitude, increasing concentrations of JWH strongly suppress synaptic transmission. So this qualitatively is very different from THC at these synapses. So these, generally, everyone we've tested, we did several, and then we kind of get tired of doing the same thing over and over again, are potent, efficacious CB1 agonists, so quite different from THC. And this is a possible explanation for the stronger effects that tend to see with these compounds and possible more adverse reactions. There's also a lot of issues about these things are probably not just targeting CB1 receptor, metabolism, purity, other issues. Next topic is functional selectivity. So this is a kind of, it's been a while since you've thought about G protein signaling. It's something that's really come to the forefront recently. So this is the idea that different agonists engage these receptors to produce different conformations and different signaling repertoires. So this is a cannabinoid agonist activating CB2, inhibits cyclase, recruits arrestin, activates MAP kinases. This other one, working through exactly the same receptor, does these things, but also does additional things. So this increases the potential richness of receptor signaling. And it has implications as well. This is work from the Halsey group. So what they've done, they're using a Huntington disease cell line model and looking at the effects of different drugs on viability of these neurons. There's a strong connection between CB1 and Huntington's that we'll talk about later. So what they're looking at membrane permeability here. So increased number of membrane permeable cells means more cell death. Down means less cell death. They find that THC and the synthetic cannabinoid actually increase cell death, whereas 2-AG, anandamide, et cetera, decrease cell death, all mediated by CB1 receptors. And when they looked at signaling, this is looking at bias signaling towards ERK phosphorylation or arrestin recruitment. They find that THC is strongly arrestin biased, where anandamide is ERK biased. And interestingly, addition of CBD to THC pushes that to an ERK bias and has similar effects on cell survival. So very, very interesting in terms of combining THC and CBD. Next, and following on that, is the idea of allosteric modulators. So drugs can interact with receptors at multiple sites. There's an orthosteric site, which is a classic site that we think of ligand binding and activation. Here, so THC for CB1. Allosteric are other sites on the receptor that may influence coupling. So allosteric modulators can affect efficiency of binding and or signaling. They can do this in a positive or negative fashion. They're often selective for the particular orthosteric ligand involved because they're maybe modulating the binding pocket size. This is so-called probe dependence. And this again enhances the repertoire, the richness of drugs affecting these receptors. So schematically, the idea is agonist binds, you get signaling. If you have an allosteric modulator, it may either enhance binding or enhance or detract from signaling. And all of these will have effects. And schematically, it kind of looks like this. If each inverted triangle here is a different, say, synapse or biological circuit, you can see with endogenous activity, you may have this effect. If you just try to block that with an inverse agonist, which is what we have in CB1 receptors, you just drive all that down just by displacing a set amount. So you can actually go kind of into negative activity levels. And that, with at least our lesson with rimonabant, is probably not a good thing. What a negative allosteric modulator will do is just attenuate all the signaling, but you'll never drive it negative. And this may actually be a beneficial thing. So it turns out that cannabidiol, our other favorite ingredient from cannabis, seems to be actually a pretty potent negative allosteric modulator of CB1 receptors. This is again work from Perry at Halifax. So what they're doing here is looking at activation of ERK on the y-axis versus increasing concentrations of 2-AG. And what they find, normally you get a nice dose response. Increasing concentrations of cannabidiol suppresses that response in a non-competitive fashion. Similarly, we found looking at the endocannabinoid synaptic plasticity, DSC, normally you see something like this. If you do the same experiment in the presence of 500 nanomolar CBD, you see a suppression. And with 100 nanomolar, you see an intermediate degree of suppression. You say, well, these are pretty high concentrations of CBD. Are these relevant? It turns out from the GW epilepsy work they presented in December, for kids receiving 5 milligrams per kilogram of CBD for 3 weeks, their trough plasma concentrations were about 40 nanomolar. Their peak concentrations were 400 nanomolar, whereas those receiving the highest dose of 20 mg per kilogram for 3 weeks, troughs of 300 nanomolar and peaks of over micromolar. So actually very, very high plasma levels of CBD, certainly within the range that we see effects here and here. So some take-home messages for endocannabinoids. So preformed precursors made on demand for the most part. Anandamide and 2-AG serve very different roles. Endocannabinoids participate in diverse physiological processes, which I just scratched the surface on. Cannabinoid receptors, THC is a low efficacy agonist, and I think these are implications for how it works and how synthetic cannabinoids work a little bit differently. Functional selectivity as well as endogenous and exogenous allosteric modulators offer both challenges and opportunities. I tend to be optimistic, so focus on the opportunities for understanding and manipulating cannabinoid receptor signaling. And a few kind of future directions to leave you with. What we really need to do is get a better understanding of the richness of endocannabinoid signaling, the relative roles of neuron glia and plasticity. Again, I've been very neurocentric here. Endocannabinoids have important roles outside of the CNS, and then how do they get around? These are lipids, they're not particularly aqueous soluble. Are they shuttled within cells? How do they leave cells? Things like that. CB2, where is it? It's still an open question. What is it doing and why? What are the therapeutic possibilities? Metabolism, peripherally restricted. I put CB1 antagonists here, but also agonists and other compounds. On the detrimental side, developmental effects, especially the effects during adolescence and the potential risk for increased psychotic disease. What are the roles, both favorable and unfavorable, of cannabidiol and other cannabis constituents, mechanisms, physiological roles, pharmacological roles? And then what is the signaling of the related receptors that these endocannabinoids can hit, including GPR55, 92, 119, and et cetera. And with that, I think I'm out of time, and I thank you for your attention. Be happy to answer any questions.
Dr. Mackie will be here all day, so if you have any questions, find them very